Dr. Baliga here. This podcast is on neutrophil extracellular traps or nets as targets and biomarkers for thrombosis. Neutrophil externalization of chromatin, referred to as neutrophil extracellular traps, has gained attention since first described by Brinkman et al. These are net-like structures released by the neutrophil, composed of DNA scaffolds coated with histones and toxic granule proteins. These have been shown to capture and disarm both gram-positive and gram-negative bacteria, adding to the neutrophil's weaponry to protect us against invading microbes. It's physiologically beneficial during an infection. It's also been shown to influence cancer metastasis. In the lungs, nets are induced by inflammation, and the tumor-boosting activity has been linked to net-associated enzymes. A growing body of evidence suggests that nets mediate the development and enhancement of invasive properties of cancer cells. A recent paper in Nature by Zhang et al. Identifies a mechanism that enables cancer cells to metastasize to the liver. Before metastatic cancer cells arrive in the liver, the neutrophils extrude a structure called neutrophil extracellular trap, that is net, which contains DNA and enzymes that kill microorganisms. Yang and colleagues report that this structure binds to a protein called CCD-C25 on the cancer cell surface. This interaction triggers a signaling cascade in the tumor cell that is mediated by the enzyme ILK and its partner, the protein beta-parvin. This pathway modifies characteristics of the cancer cell and alters filaments in the cytoplasm that affect cell shape. This change that occurs increases the cancer cell's adhesive and invasive properties and boosts its proliferation. Future studies will be needed to assess the feasibility of targeting CCD-C25 for anti-cancer therapy. In addition to its role as adding to the neutrophil's weaponry to protect us against invading microbes, uncontrolled and excessive net formation may however, be harmful by enhancing thrombosis. Other cells, such as eosinophils, basophils, mast cells, and macrophages, are also capable of releasing extracellular traps. Numerous studies have described direct and indirect mechanisms by which nets enhance thrombosis. The prothrombotic role of nets was shown by Fuchs et al. The study showed that nets capture platelets, promoting platelet activation and aggregation. Nets have also been shown to promote thrombin formation by providing a scaffold for red blood cells and procoagulant molecules such as 1 Willebrand factor, fibronectin, fibrinogen, factor 12, and tissue factor, as well as procoagulant extracellular vesicles, including tissue factor bearing extracellular vesicles. As such, the histone DNA backbone of nets is thought to add stability to the fibrin scaffold in thrombi. Moreover, fibrinogen clotting in the presence of histone DNA complexes results in thicker fibrin fibers, a higher fibrin clot stability and rigidity, as well as significantly prolonged clot lysis time. Despite a large amount of research on nets, there are still no standardized methods to assess net formation, and caution should be taken when interpreting data on net formation. Potential assays include various enzyme-linked immunosorbent assays such as nucleosomes, neutrophil elastases, myeloperoxidases, myeloperoxidase DNA complexes, and citrullinated histones, ELISAs for nucleosomes, neutrophil elastase, and myeloperoxidase, however, do not specifically determine net formation. In addition, there are problems with standardization of myeloperoxidase DNA and citrullinated histone 3 ELISAs. Flow cytometry assays detecting net components on cells and extracellular vesicles have also been proposed as rapid assays for the assessment of in vivo net formation. Various net components in plasma have been proposed to predict disease severity in coronary artery disease and ischemic stroke. For instance, plasma levels of nucleosomes and myeloperoxidase DNA complexes are associated with coronary atherosclerosis severity and ischemic events in patients with coronary artery disease. Plasma levels of extracellular DNA, nucleosomes, and citrullinated histone 3 also correlate with stroke severity and outcome in ischemic stroke patients. Plasma levels of circulating extracellular nucleosomes and neutrophil elastases are associated with a 3-fold increased risk of DVT in a small study of 150 patients. Similarly, increased levels of circulating extracellular DNA and myeloperoxidase have been shown in DVT patients compared to symptomatic DVT negative patients. Several studies have examined the role of NETs in cancer-associated thrombosis. Although there is data showing increased susceptibility of in vitro net formation in cancer, the in vivo contribution of NETs in cancer-associated thrombosis is less well studied. In light of the pathophysiological relevance of NETs, the development of therapies blocking NETs is ongoing. For example, the inhibition of peptidyl arginase deaminase 4 has proved to be effective in a variety of conditions such as stroke, acute myocardial infarction, and diabetes. Besides, Janus kinase JAK1 and 2 inhibitors that lower net formation in mice and humans has been shown to experimentally reduce thrombosis. The negative side of these potential therapeutic pathways relies on the non-uniqueness of the targets acting also in other processes than netosis. In thrombosis and cancer thrombosis, biomarkers and therapeutic targets of NETs is the next frontier in research.